Hello, a warm welcome from my side. My name is Dürkop. I'm the CEO of Novasign GmbH. We're a spin-off from Boku University, and my talk today will be about hybrid modeling and intensified design of experiments. Basically, I'm currently in my home office, but I dressed up for you. Maybe you see I have a nice view to the outside. But okay, so let's directly jump into the topic. Actually, right now we have a huge crisis, Corona crisis, and one of the problems we encounter right now is that there might be a vaccine on the market in 1 to 2 years. But normally, when we talk about medicine development for the market, the normal timelines are around 10 to 14 years, which is a huge time span. And the development process itself consumes something like $2 billion. That means once such a drug is on the market, it comes up with quite a huge price tag per dose. It is, at the end, sets huge pressure on the patients and also on the health system. So I think we're in a stage where we really have to speed up this process. And in order to speed up this process, we have to consider actually what are the bottlenecks. So when you look at this simple sketch, you see that from a new molecule or a biosimilar, there are actually two streamlines at the same time. The one side is the bioprocess or the process development, and the other side are the clinical trials. And as I said, this time span is still 10 to 14 years. And we have to ensure, or we have to find solutions to speed up this process. Okay, so our aim is to significantly reduce both the process development and also the clinical trials. So from the whole perspective of mankind. And this will have two benefits. On the one hand, we would have earlier patients treatment, especially now if you consider Corona. I think this is quite clear. But also it would lead to significantly higher return of invests for companies. Because if you enter the market earlier with your biopharmaceutical or with your drug, you have a longer time span until your patent protection runs out, and you can supply the market alone. So this is also a huge benefit for companies. So we have to reduce both of these timelines simultaneously. In order to do so, you have to somehow consider the value chain, and the last point in the value chain, the last point is actually the patient. So the patient, if you consider Corona, has a problem, and so has a request, and we need a drug to treat this patient. And on the other hand, you can think about that the drug or the product must be fit for the patient, and you can consider this something like to be the clinical trials. So you have to make sure that the product fits the patient. And but this continues. Also, the product has to fit the process, and finally the process has to fit the plant. So something like the inverse value chain. And currently there are a lot of initiatives speeding up this process from different perspectives, and we call this in the frame of digital transformation or digital twins. And for all of these particular parts, there's actually a lot of development to deliver digital twins. So you can consider like patient data, you can do a lot of patient data, you can think about molecular modeling, you can consider models about the process, but you can also consider these building information models where a lot of different vendors can work together in a single plan and can deliver the construction of a plant much faster than it was before. So there are actually a lot of tools around the market to speed up all of these different topics. And you can, for example, think about Google DeepMind. They are really strong in machine learning from patient data. But you can also consider what we are doing, and what my talk will deal about today is about the relationship between the product and the process. And we use hybrid modeling here to speed up the process development. So we are only on the process development side, or on the process side and not on the clinical side. But you can also think one step further, and you can say, okay, there are also tools out there. I mean, this is not a comprehensive comparison, but there are just 3 tools to give you an idea of what's out there and on where these tools address on different levels of the process. You can also consider like what Siemens is offering. Siemens, it's an engineering plant simulator, which might help you with digital commissioning, and you can also speed up timing of plant construction. Okay, so coming back to what we are doing with Novasign, we're into process modeling. So when you consider there's a certain there's a certain cell, and from the cell there's a certain process, and at the end of the process there's the medicine. And what we are doing is we're speeding up this process using advanced machine learning tools. When we look at operations which are currently out there used for biotechnology, I would say there are three main topics. There's the upstream topic for sure. If you have a bioprocess, you have to produce this somehow in cells. So upstream processes everywhere. Second thing is filtration, which in every process always there's at least one filtration step, either for separation or for concentration. And finally, also chromatography. In red biotech, we also always have chromatography. In white biotech, it's not so often, but still over the whole frame of biotechnology, I think these three unit operations are driving really the market. So, and today I want to show you two showcases. One is for the upstream bioreactor and one is for the filtration process. We start with the upstream bioprocess. And here I have a showcase from an E. coli fed batch cultivation, a 20-liter reactor. And what we were interested there is we had three critical process parameters: temperature, feeding rate, and induction strength. And these three parameters were supposed to drive the target parameters, so the critical quality attributes, which are in this case the biomass and the product. So if you look at this design space, it's quite clear that it is an immense experimental effort, because each of these points represents one experiment, and one experiment here in this E. coli cultivation is 1 week. Not as long as CHO, but still, if you do all of these experiments, that means you have to do 27 experiments, which cost you at least half a year of experimental work to understand the whole design space in the full factorial way, okay? What people are currently doing, or often doing, is they take ordinary DoE tools, so they have the design space, then they perform their experiments, and then they look at the endpoint. So the endpoint of the process, then they build a model. You can use Jump, SunExpert, MATLAB. There are a lot of tools actually. You can build a model to describe the relation between the inputs and the outputs, often for the endpoint. But there's actually a limited process understanding if you only look at the endpoint and you do not model the whole process in a time domain. And as an example, you can see this here. So if you have the response variable, might be the titer over the process time. And let's see, this is a comparison of 4 different runs. If you look at the endpoint, there are actually infinite possibilities of the trajectories, how you get from the start to this endpoint. So let's say for this particular run, you might have these three options. And if you do not sample over time, and in the next step, if you do not take a model over time, you have this somehow, if you do not model over time, you have this somehow called DoE knowledge gap. So if you model over time, let's say this is the true run, you know, okay, this is the trajectory, this is how the process develops. And if you look at this over time, the first thing you will realize is, okay, there's no need to have this process for this particular time, because if you stop earlier, you might have the same process yield at the end, but the time space yield is much higher, because you don't have to perform the whole run. That's the first thing. And the second thing is, when you also take samples over time, you can really build up a very valid process model, which describes the whole process. And this, in our case, was done by hybrid modeling. And you can see this here. So we have three input parameters: temperature, feed, and induction. So there's a little thing not right, but never mind. We have these three inputs: temperature, feed, and induction. And these three inputs, they are used in an INN. And this artificial neural networks then predicts two rates. The first rate is the growth rate, the mu, and the other rate is the product formation rate. And by doing so, we feed these rates into a mechanistic model. And this mechanistic model gives this whole model a certain structure. And this is why it's called hybrid modeling, because we have the INN, which is a black box model or a data learning model. And the other, the white box, is driven by differential equations here. And as the output, we have two output variables. One is the biomass and the second is the product. Okay. And by doing so and comparing the outcome of process modeling from hybrid modeling with pure black box modeling over the whole time domain, you can see here in a simple example, for example, you see the biomass prediction and a scatter plot for the pure data-driven model. And what you see is on the x-axis basically is the measured value, and on the y-axis you have the predicted value by the model. So if you would have a model that perfectly fits all of your data, you would have a straight line. However, we also know that we have a certain analytical impreciseness, so we don't know it. There's an inaccuracy in the measurement. We know, for example, for the biomass, we did this in our experiments where we performed 7 rounds the same way. And then you can see for each of these points, you can measure the biomass and you can see how much they deviate. And in our case, something like 5%. So you can see that what you can see from this comparison between the pure black box model and the hybrid model is that the hybrid model, due to this given structure we introduced by the mechanistic equation, the predictability of the outcome is much higher. And you can also see this in terms of the time domain. So while in the upper graph there was a scatter plot, you see when you take one of these runs out of the scatter plot and plot it over the time domain, you can see, for example, how the pure black box model performs over time and how the hybrid model performs over time. And what we also did is we aggregated several models. That means we did a bootstrap approach. So you take several models and you take the average of these models. And then this average of this model gives you somehow confidence interval of the model. So it's not a real confidence interval, but it says, okay, how many different models you have and what do these models predict? So if this confidence interval of the models is quite broad, that means all of the models are quite different. And if this confidence interval is very small, it means your models are quite similar. And not only that the hybrid model can predict the trajectory of the real biomass samples, which are obviously these points here, or these triangles here. And so not only you can predict better the real trajectory, but you can also, all of the models, or the different models, they are much more similar than for the pure black box model. So this is one of the learnings here. But right now, what we did so far is we were able to describe a process, which is nice. You were able to describe the whole process, but you didn't save any time. So my initial argument was to save time, and this is what we really want to do. Right now, we just described the whole process, but hybrid modeling in this case here is like the commodity. So you have to describe the whole process first, and then when you're able to do this, or if you have a tool that is able to accurately describe the whole process, you can go one step further and really save time. And this is what I will show you now. We have the approach of intensified design of experiments. So what does this mean? When you have a full factorial design space, what I told you before is these 27 experiments. Each of these points represents one experiment. We have 27 experiments to conduct. It means half a year of work, okay, to screen the whole design space. But what you can also do is you can take a fractional factorial design space. That means you take out some of these runs and you only perform, in this case, only the borders, only these 4 points in the top and the 4 points in the bottom plane and the center point. So that means by doing so, you save 66% of the experiments, and the questions you learn the same than if you would take the 27 experiments. And this is what basically is done in industry also quite often, because if you consider a CHO fermentation, for example, where you have 6 to 7 parameters you change on 3 levels, so you have 3 to the power of 7, you cannot do a full factorial design space because this would take infinite time. So you have to take points out and you have to reduce the number of total experiments. You can either do this with the fractional approach, but what you can also do is you can consider this intensified design of experiments. What you do here is you try to still do all the experiments in the design space, but not each point represents a whole run, but you change process parameter during the cultivation. So what you can see from the blue plane, for example, is all of the 9 points in the blue plane are covered by one of these lines. So a line starts in a certain point and then covers 3 of these points. That means you start with a single condition, and after a certain time, in our case, we change after one doubling of the cells, we switch to a new cultivation condition, and then we switch again to new cultivation conditions. So in our case, we did 2 switches for each of these EDRE runs. And if you do so, you also have only 9 experiments. So that means you also reduce the total amount of experiments by 66%, but you somehow still learn from the same design, from the whole design space. This is called intensified design of experiments. Then you can make a comparison. For example, if you compare the 3 tools, the DoE, only at the endpoint, because we just look at the endpoint for this particular thing now. We only look at the endpoint for DOE. We compare the model predictability by taking all the points in the hybrid model and also doing hybrid model with this intensified design of experiments. So while we have in the DOE 31 experiments in the training set, in the hybrid model we took 25 in the training set, training and validation set, and 6 in the test set. And in the hybrid model we only have 9. And for test the hybrid model, what we did here is we took the hybrid model from the intensified runs, so the 9 runs, and built a model and tested this model then on the 20 or on the 31 experiments. So the reason why here 31 stated is that we performed some of these experiments as duplicates. So therefore we don't not only have 27 experiments, but the full design space was 31 experiments, but still covering the whole points described here. And as I said, what we did here is we performed the hybrid model on the data from the EDOE. So we built the model on the IDOE data. And then we took this model and predicted the concentration for the biomass in the product on the statically design of experiments, so on the full factorial design of experiments. And when you look at the data, you see that the normalized root mean square error is lowest for the DOE point, but this is also clear because you only look at the plane, you only look at the endpoint. For the hybrid model and the EDOE, we are able to describe the whole process. So the error is a little bit higher, but you can describe the whole process. It's the first thing. So you have an entire process model. It's a huge advantage. And second, when you look at the time, or if you look at the required experiment, and this represents the time and also represents the cost you have to do in your lab, that means you can significantly save time here. And finally, what you can do with the whole process model, you can also use it for soft sensing. So you can implement soft sensors, and I will show this to you afterwards. You can implement soft sensors, and in the next step, you could also go for advanced process control. So you have a model, you build the model on the design space, you learn from it, and then you can also transfer it to the shop floor. Or if you are already doing verification runs on the shop floor, you can also use these models for soft sensing in real time. Okay, so this is for the biomass, but you can also look at the product where we have a similar picture. However, the normalized root mean square error is a little bit higher. This also comes from the fact that the analysis is not so accurate compared to the biomass analysis here. But apart from that, you see a similar picture. Okay, then let's go to one of the examples just for a single run. So here's just a single run over the time domain. We compared the 3 different outcomes from the hybrid model built on the full factorial design space, on the hybrid model built from the intensified design of experiment, which is the green one, and the hybrid model built only on the fractional design space, which is the red one. And you can see that for sure, the hybrid model built on the full factorial design space, which is the blue one, performs best. For sure, you learn from the most data, you have the most data to learn from. But you can see that the IDOE model, which learned from the 9 IDOE runs, also is quite accurate, and also the bars of the model are not as broad as for the fractional factorial design space. And the huge benefit is, so you learn from the whole design space, you take up the whole dynamics from that, and you save 66% of the time, which is a huge benefit. Okay, coming to the soft sensor application, as I said, we just built this model. We just built this model based on these EDOE runs, and you can transfer it back, and you can say, okay, why not doing something different, or why not performing experiments in the lab or on the shop floor, or wherever you are, and use these models as soft sensors so that you have a real-time prediction of your biomass and your product during the new cultivation. Okay, and this can be done quite easily. So we have a hybrid modeling toolbox. We build this model inside our hybrid modeling toolbox. You can export this model as a DLA file, and then you can import it at your environment which you are using. In our case, we're using Evong Sum Control, and we are importing in Evong Sum Control the DLA file from our toolbox, and I can show you afterwards how this soft sensor performs. Okay, so this is the slide of the soft sensor performance. Normally this would be a video, however I cannot play this video here, but I have a QR code for you. So if you're interested, you can just scan this QR code with your phone. You will be directed to YouTube, and on YouTube you can see how this propagates in time. So this is a prototype. what you will see is, for example, here you have the temperature. So this noisy signal, here's the temperature, and here we have this shift. That means this is an IDO rerun. We perform the new IDO rerun, and you can see, for example, here the temperature shifted. That's why we have this here. When you look at the curve, the red curve, for example, would be the biomass. You can see how the biomass propagates over time. And the yellow curve, which starts here at 4.5, would be the product, which would be which also grows over time. And you can also see how the product propagates over time in real time in the facility. So this is the soft sensor in real time. Check out the video. You will see at the beginning of the video how the DLL is imported, how the model parameters are mapped with the OPC parameters. So the three model parameters we have are mapped with the corresponding OPC parameters in the environment. And then we just start the cultivation, and we see a time lapse how this propagates over time. Okay, however, I also have the outcome of this for you in a simpler slide. As I said, we performed the EDO runs on this design space, and then what we did with the soft sensor here, we performed a complete new run. So we did a different IDO run. We started with this condition. After one generation, we switched to this process condition, and after another generation, we switched to this process conditions. Okay, you can see this here. So while you have the prediction of the soft sensor in black, this is what the soft sensor predicts in real time. You can see what the offline measurements afterwards in yellow would give you the real numbers. These are the real numbers compared to the soft sensor numbers. And the advantage is obvious. While the soft sensor is in real time, you have this number right away. The yellow real-time measurements come in 1 to 2 days to date. This is the first thing, and the second thing is that the soft sensor, how it is built, is also a predictive soft sensor. That means you can simulate, because our input is the feed, the temperature, and the inductor, and you can simulate how much you would feed into your process in the future. That means you also get this curve already for the future. This is also a huge advantage if you build models with a valid model structure, and the inputs are factors that you can control. If you take an input factor, for example, from some spectroscopic measurements, you have other advantages, but the disadvantages you cannot look in the future because you don't know what the signal will be in the future. And also, when you think about predictive control, you also cannot change the signal of a parameter, for example, in order to get more product out of it. You can only do this if you build your model based on variables you can control. Okay, here you can see the same for the product. It's also quite accurate. However, we have a deviation at the end of the process. We cannot really say if this is either from model inaccuracy or there was something wrong at the end of the process. But still, you can see also for the product that the soft sensor is quite accurate. Okay, so summing up the upstream part, this hybrid model is more suited to describe bioprocesses on DOE through the Blackbox modeling. Second, the IDOE significantly saves process characterization times. And thirdly, I hope I could show you a simple implementation of a soft sensor at the shop floor or at the floor or wherever you want to apply it on. And it can be easily used to access both the biomass and the product in real time. In this particular case, if you have other process variables, if you have other quality attributes you're interested in and you measure them, you could also predict them in real time. Okay, now coming from a different topic, now I think I could show in the upstream topic why hybrid model is very useful there and how this could speed up your process. The second thing is now the downstream showcase, where it is about tangential flow filtration. Just as a short reminder, what is tangential flow filtration? You have a certain target of interest in a certain in your tank, and you concentrate this in. That means you pump your solution through a membrane cassette, and everything that is smaller than everything that is larger than the pore size of the membrane cassette stays inside this flow, and everything that is smaller just goes through the membrane. That means you concentrate your protein solution in, and this is a time-depending process. And there are a lot of parameters that are responsible for this permeate flux. So the permeate flux is what goes through the membrane. And in our case, we build a model based on the cross flow. So the cross flow is one input parameter, the transmembrane pressure is one input parameter, and the target of interest is for sure one target parameter. Okay. So here we go. And the objective of this is to have a prediction of the duration. So we want to have a prediction of the duration, how long this process might last under certain conditions. So let's say we have a certain TMP, a certain concentration, and a certain cross flow. How long would this process last? And for different concentrations, so let's say you have a certain variation in your process, plus minus 1 gram coming from a certain column, then you also have different outcomes. So if you have a model that can describe the duration under certain conditions, you can take action and you can change them. So we want to build a generic model. The second thing that is usable, that can be used in different operation mode. I will explain what I mean with this afterwards. And developing a digital twin is the third point. Okay. So the hybrid model structure here is somehow similar to what we saw before with upstream. We have 3 inputs, TMP, CF, and concentration. We predict the flux, and the flux then goes into a mass balance, and from this mass balance, we calculate the concentration at a certain time point. And we take this concentration, put it into a multi-step hybrid model, and do the same thing again. And by doing so, we could look into the future how your process will develop, and I will show you this in this sketch. So what you will see here is you have the tank where you have your protein of interest. This is the yellow thing to the left. And when we start the process, this volume will decrease, and also the flux will decrease. So when we have low protein concentration with the high flux, and the higher the protein concentration side your system gets, the lower the flux gets. And you will see, so for example, what you see right here, this is the outcome of the model. So you see what your model will predict under these conditions for the future, how long this process will take, in this case 70 minutes or 50 minutes, and then you start the process. And what you see now is the real process. So here you see the real data, and this is how real data propagates over time. Exactly. So you see that the model is quite accurate, and it's easier than in the upstream because the uncertainties are not so high than in the upstream. The things you're looking at and the model parameters which shows here are really almost 100% describing the process. Okay, so this was for a fat batch cultivation, but you can also use this model with low error here. Exactly, you can also use this model for different unit operations. Here comes the power of the hybrid model, because you can also use it for fat batch TFF. What does this mean? Well, in a fat batch TFF, this is something that is typically often done in large scale, because you have a small tank and you have a large surge tank where you have a higher volume in there. So overall, the volume is too high for the small tank. That means you concentrate in the tank, and the volume that goes to the permeate is filled up again by the surge tank. And by doing so, you see a different behavior. So you see this up on the upside here. Upright, you see that while the surge tank, there's still volume in there. You see quite almost, let's say, a pseudolinear line. And once the surge tank is empty, you can see that there's a steep increase or a decrease in permeate flux. So you can see this here, how this behaves over time. And the power here is that we learn from the data, but due to the fact that we build a hybrid model, we are able to predict both the batch TFF, also the fat batch. What we're currently working on is, this is the second part now. And the second part now represents this steep decrease in the permeate flux. You can see a similar error here for different process conditions. Okay, but when you have this hybrid model that describes the relationship between your membrane and your protein, you could think about different unit operations. So you have different operating modes. You have the batch mode. You can also think about the fat batch mode. But since you know there's behavior between the protein and the cassette, you can also think about parallel SPTFF or serial SPTFF. You have to change some things in the mass balance or in the white box, but still, due to the fact that you learn from the behavior how your membrane reacts to the protein, you have a very high learning rate, and you can extend this model on different operations modes, what would not be possible with the pure black box model. Okay, finally, we also have a digital twin application here, and this is what I said before, due to the fact that we learned from the behavior of the membrane to the protein. We could also model different situations. So let's say, okay, now you have a different concentration, starting concentration, but you want to keep the process still at 40 minutes. If this is a time slot you're interested in, you can consider how you have to change the TMP and the gross flow in order to finish also after 40 minutes. And you can see a simple example in this sketch. So the algorithm iterates through. So let's say we have a 1 liter reservoir, and we want to finish after 30 minutes. So the algorithm iterates through until it finds the optimum conditions to stop to be finished with a certain volume that you set up front, be finished after 40 minutes. And with this digital twin application, you can really model your process in order to to deal with different situations. So if you have, as I said, different situation where you have a different protein concentration at the starting at the start, or if you have a different volume at the start, you can all model this very simply with this approach. Okay, so this brings me to the results of the downstream topic. Is we have a multi-step ahead hybrid model that can predict the outcome of this process already at the very beginning of it. It's a very small training set is only sufficient, so you can do normal TMP excursion as you are used to in industry. The only thing you have to consider is that you need a device that is able to keep the TMP, has a TMP control, because if you do not control the TMP at the end, it's clear if you have two variables that you don't know, you cannot model this process. So you have to have a system that is able to keep the TMP constant over time. Okay, and then the flux evolution over time was modeled, and ideal time point for DF, for example, could be estimated. So when you say you know the ratio where you want to change to DF, then you already can describe this outcome. Okay, and also the between application, what I showed you last is that with this simple thing, you can upfront 5 minutes before you start your process, you can set the ideal conditions you want to have in the process, and at the end, it will lead to your desired outcome. Okay, last thing, some advertisement for our toolbox. We are currently developing a hybrid modeling toolbox, which could be used by every one of you. If you're interested, and just go to our homepage, novasign.at, and you can download this toolbox for free. So there's a free version. Basically, it's a freemium concept, so you can always use it for free. There's a limited version can be used for your process. Mainly, we're focusing on upstream processes, but currently we're also integrating what I just showed you in the downstream, which is currently still in MATLAB inside our hybrid modeling toolbox. And we also have support portal where you, on the one hand, can track your own progress, and on the other hand, you can get support from us if you have any questions. Now, with this, I'm at the end of my presentation. I'd like to thank you. If you have any questions, I think my email address will be given to you, so you can always contact me, or you can use our channels like LinkedIn, Twitter, or direct mail. Maybe if this is a responsive slide, and you can also just click on this slide. Okay, with this, I'm at the end. Have a nice day and stay safe. Bye.